One of the great things about having all this data integrated in the BV-BRC resource is that you can look at different things without leaving and look at your experimental results and quickly find things downstream of interest. So in this particular video, I want to look at where the SNPs are on a protein structure. I've done this before in the multiple sequence alignment videos, but I want to show you a little bit of a different flavor to it here. So we're working with Streptococcus pneumoniae. So first I'm going to look for the taxon and find those structures. So I input the genus and species name, and then I go under taxa to the genomes. So I click on that, and there are 30,161 genomes. I want the structures. So I click on those, and there are 2,309 structures here. Now, there's going to be a lot of stuff that we're not interested in, so we can filter. We're doing the PBB2X protein. And notice when I put that in, there's often a lot of noise here. So let's see if I can sort on it by 2X. And sometimes you can also see it in the name here. You know what? I want to create some real estate here. Look, these are all 2X. As well. Let's see. Here is one that's interesting. So if we look beyond the green bar, this is the penicillin binding protein 2X from Strep pneumoniae in complex with oxacillin. Oxacillin is commonly used with Streptococcus pneumoniae to do a quick test to see if it's going to be resistant or susceptible to beta-lactam antibiotics like amoxicillin, like penicillin, like ampicillin. So this is an example of that, and I want to just see what it's like with that. So let's look at the structure. So I click on that. Oh, good. Okay, so here it is. One thing that you can do is I could click here to see the ligand, and that should show me the oxacillin. And here is the beta-lactam ring here in this, and you can twist this and you can see how it's referenced here. And if, for example, we know that one of the most important mutations is at 339, you see how it's pink there? Let's see if I rotate it a bit. Let's go to 339 again, where it is close to that protein. Another one of the proteins that are important is 371. So I like how it's showing me at the bottom when I'm close. And so that's close too. Then 384 right there, but I don't see it in line, but you can twist this thing around to try to see these things. Okay. So there's this view of it, but there's also this view, which I like. And if I twist it around, you can see that the oxacillin is right in there embedded in this protein. So this is the illustrative view. And let's see if we can tease out 339. Oh yeah, you can see it barely. You can see that structure. It'll be right in this area when I mouse over it, but you can see it right there. Do you see how it is? So it's buried more deeply in the structure. And then there was 371. Try to see where that is. And 384. That's farther away. 400. Oh, that's actually pretty close. If you can see, it's lighting up right in here. So 400 would be right in there. Oh, this is fun. I generally use the default as opposed to the illustrative, but I do like this. 514 and 546. 514, well, that's pretty close too. It's hitting right around in here. And then 546. And that's close too. And then 605. Okay, that's further removed. I like this better because it's showing you a different view of it. And you could see that here's where the antibiotic is bound. And if you recall, the transpeptidase, PBB2X, forms this pocket that the antibiotic fits in. And you can see it's fitted in right there. And when you look at it with this view, you can see that these amino acids are in close proximity to this most active part of the protein. So I just think that's really exciting, and have fun with it. Don't just stick with the default view. Go ahead and go to the illustrative view, and you can quickly find things that are in the view by clicking on ligand. Will show you that, and it also at the bottom will tell you what this is when I click on it. So that's the oxacillin within the molecule. So have fun with it. It's really great to give you a sense of where things are happening. And when you're lucky enough that there's an available structure with a closely related, you may not have the antibiotic that you're most interested in, but a closely related one, and then you can see it and see where these crucial amino acids are in the context of the drug. It's really exciting. So give it a try and have fun. See you later. Bye.